I'd like to extend a warm greeting to all of our alumni, students, colleagues and friends who are watching from across the globe. My name's Craig Rutherford and I'm a Development Officer in our Development and Alumni team. If this is your first Science Talk, then thank you very much for joining us. Our Science Talks are short presentations by leading members of our academic community on key areas of our world-leading research. It's a little flavour of the expertise our lucky students get to see on a daily basis. This evening's talk will be given by Professor Malcolm White from our School of Biology. The talk will last for about 30 minutes and then Professor White will be happy to field your questions. We'll aim to wrap the event up by about 6:15 at the latest, so do please get your questions in early if you'd like to ask anything. If you'd like to ask a question, then they can be added to the chat bar on your screen. Before we get into it, I'd just like to do a quick plug for the next talk in our series. We've got a varied schedule of talks extending throughout the academic year, and the next one will be presented by Professor Julia Priest from our School of English, and it will be on colonial era Caribbean theatre then and now, and that takes place on Wednesday the 15th of November at the same time. You can also access the full archive of our past talks on a plethora of subjects from bird migration to tackling coronavirus on our dedicated St Andrews Alumni YouTube channel, where this talk will also be available over the coming days. Before that though, let's get back to this evening's topic at hand and tonight's guest. Malcolm White was appointed as a lecturer at St Andrews University in the year 2000, following periods of postdoctoral research in both Scotland and California. He's interested in molecular evolution and studies bacterial immune systems, discovering new enzymes and pathways that provide antiviral immunity by detecting and destroying viral invaders. Malcolm is funded by the European Research Council and is an elected member of the European Molecular Biology Organisation and the Royal Society of Edinburgh. Malcolm's talk this evening is on the bacterial roots of the human immune system. All living things since the dawn of life have been attacked by viruses, which are parasites that need to take over host cells to replicate and to spread infection. Cells have developed many forms of defense against viruses, and viruses in turn have found ways to overcome these defenses. Although arising from fundamental blue skies research, the discovery of the bacterial CRISPR immune system 15 years ago offers a way to precisely edit the DNA of any living thing and has already revolutionized many areas of research and biotechnology. By studying the systems bacteria use to combat viruses, we are now realizing that humans inherited many of our antiviral immune systems from our bacterial ancestors. This talk will explore the link between all living things on the tree of life, with a particular focus on our shared immune systems. So without further ado, I'll hand you over to Malcolm for this evening's talk. Malcolm.
Thanks very much. Thanks for the introduction. And yes, as Craig said, I've been here in St Andrews for the past 23 years. Time flies. Based in the Biomolecular Sciences building on the North Hall in St Andrews. And my lab studies bacteria and the way that they protect themselves against viruses and also against damage to their DNA. So today I'd like to take you through a little tour of some of the aspects of this that are of particular interest to me and hopefully to you as well. And then I'll finish the talk by providing an update on some of the very recent research that we've done in this area. I want to start by considering some trees. As we've seen recently with the Sycamore Gap, trees are very important, both as symbols to us. And this has been true throughout civilization. So if we look at this Renaissance painting by Hugo van der Goes of the Fall of Man, we can see central to this is a tree. This is, of course, the tree of knowledge, and you can see Eve there reaching out for an apple from the tree. In a way, I think Eve is therefore the first scientist searching for knowledge and grasping it. Meanwhile, Adam looks on. He's got a very perturbed expression on his face. I think he's maybe thinking we need to get some better PPE. And the serpent here is an unusual creature. Of course, it still has arms and legs at this point and looks like an early example of genome editing, to my mind, which we'll come back to. So trees have been a useful way to think about science, and that's perhaps personified by this one of the most famous trees that has been reproduced on many a t-shirt and mug, which is one drawn by Charles Darwin in his notebooks when he was thinking about evolution and speciation and how all living things are related to one another. And he used this tree as a way to help him think about how evolution works. And it's a very powerful symbol that is still at the basis for a lot of scientific thinking. We are now able to sequence the DNA, the genomes of any living thing, relatively easily and cheaply and quickly. And that allows us to build much more complex trees and try to represent life as a tree where we use molecular information. And that's what's shown in this tree. This is a version of the tree of life from earlier this year. It was created by a colleague at the Pasteur Institute, Simonetta Gribaldo, and her student Basil Bode. And in this tree, obviously, there's a lot more information here. And it essentially shows a representation of the diversity of all living things. Now, I know you're not going to be able to read the details, but essentially, you will see almost two halves of this tree. One half is dominated by organisms called the bacteria, and the right-hand side, organisms called archaea. Now, these are both single-cell microbes, and they predominate much of life as we know it. On this tree, then, the eukaryotes, which are where we are a member, the eukaryotes are organisms that have a nucleus. That includes all the animals, plants, fungi, etc., including humans, are present as quite a small part of the tree. So we might think we're the top of the tree, but we're actually just a rather minor branch on this tree. And we're represented here by a little image up here of Carl Woese. Carl Woese was the scientist who first started to use genetic information to try to relate all living things on a tree of life. And he was the discoverer of the archaea, which are a fundamental part of this tree that were only really recognized maybe 40 years ago. So let's just simplify this tree a little bit by looking at the roots. And I want to just, so there we are there, sorry. You didn't see Carl Woese up on that corner. I want to start by thinking about how life evolved at the earliest times. And so what we think existed before the diversification of this tree was an organism called LUCA. LUCA stands for Last Universal Cellular Ancestor. And this reflects the fact that all living things are fundamentally related to one another. They all use DNA, for example, and they all have the same apparatus to replicate DNA, to read it and to make copies of themselves. So LUCA at the root of the tree, then life diversified, and you have, as I said, the bacteria here on the left, the archaea on the right. They split very early, probably quite early after the planet became a stable biosphere for life, which is probably roughly when liquid water began to be available. And so as life diversified, the bacteria and the archaea went their own way, and the new species and developed. And at some point, a couple of billion years ago, the bacteria and the archaea fused together. So there was a fusion. That may have been an archaeal cell swallowing up a bacterium. And that is thought to have created the third form of life, the eukarya or eukaryotes, of which we are a member. And so in the eukarya, the bacterium has become what we call a mitochondrion. That's essentially a power pack that generates ATP as energy for the cell. And this fusion allowed a profusion of more complex types of life forms, of which we are one example. So what this means is that although we really consider ourselves very complex organisms and more complex than bacteria, we carry the original components of the bacteria and the archaea that denote our sort of ancestral past. Now, I want to talk a little bit about DNA. So almost all living cells are absolutely packed full of DNA. And as you can see, this bacterium, which has been burst open, and the DNA has been stained, you can see how it's all spilled out. And probably looks like there's more DNA than would fit in the cell. And as I'll show you, it's really amazing how cells can pack in a large amount of DNA, which is needed as a sort of recipe book, the instructions to create new copies of themselves. And yet, although this DNA is wrapped up tightly and packed, it has to be manipulated, it has to be read, it has to be replicated in order for life to continue. So I want to just spend a little bit of time thinking about DNA and putting it into context when we think about scales in biology. So in biology, we're quite used to thinking across a lot of scales of dimensions. So some of us molecular biologists might work down at the nanoscale. So this is a scale in meters. So nanometer, 10 to the minus 9 meters. That's about the size of a simple molecule like glucose. Proteins, which we study and many people do, are maybe 10 to 100 times bigger than that. And then the viruses start to come in around 10 to the minus 7, 10 to the minus 6. That's a micron. You'll find bacteria around about a micron in size. Organisms like E. coli, which we use in the lab as a tool. Red blood cells a bit bigger, and then the first multicellular organisms, like this is a nematode worm, about 10 to the minus 4 meters. Working your way up the scales into up to 100 meters, maybe that's roughly the size of the biggest living things like whales and trees. So biology, different parts of biology work over these different scales. And I want to now place DNA into this representation. To do that, I need to expand the scales a lot, actually. So let's start by thinking about the structure of DNA as a molecule. And here we're on the nanoscale. So DNA is formed of steps called base pairs, and each base pair carries a tiny piece of information. And each base pair is important for the information to create a new organism. So that's down at less than a nanometer, 0.3 nanometers distance between a base pair and the DNA. However, you may know that the human genome consists of about 6.4 billion of these base pairs. So almost all the cells in your body have about 6.4 billion base pairs. And you can do the maths here and work out, well, what's the length of DNA in a cell? And it's quite mind-boggling. So in a normal cell, there's something like 2 meters of DNA. Now, a cell is really tiny. You need a microscope to see a cell, and yet there's in there 2 meters of DNA. It's very tightly wrapped up in a very sophisticated way to allow it to fit in the cell, but still be useful and still be replicated. So if most of your cells in your body have about 2 meters of DNA, then you can think, well, how much DNA is there then in total? And we can roughly calculate this. There are some approximations here, but roughly, if you think of the number of cells in the body and each one has 2 meters of DNA, then in a human body like yours or mine, we have something like 10 to the power of 12 meters of DNA. That's a huge number. It's a very hard number to really get your head round. So we need to go into astronomical kind of comparisons. So the sun is about 1.4 times 10 to 11 meters away from the Earth. And that means that the DNA in your body, if it was stretched out, would reach to the sun and then back to the Earth and then back to the sun, back to the Earth, back to the sun, back to the Earth. Okay, that's roughly the amount of DNA that you have and I have in our bodies. So that's quite an astounding amount of DNA. We can actually take it one step further and think, well, in a human lifetime, how much DNA is there in one human life? Because of course, your cells replicate, they die, they're replaced, and the human body you have at the end of your life is completely different from the one you started with. And again, there are some approximations here, but you can calculate something like 2 times 10 to the 15 meters of DNA in an average human lifespan. Now that's getting close to light years. So we're getting into light year territory. The nearest star other than the sun is about 4 times 10 to the 15 meters. So a fairly average life may be making close to light year of DNA. And remember, although that's an astronomical unit, here we're talking about a very information-rich molecule. Every step if you think of DNA as a ladder, and it might be nearly a light year in length by the end of a human life, then every step is still only 1 nanometer apart. And every step's important. There's information in there, and that DNA has to be protected from damage. DNA is a molecule. It gets wear and tear. It gets damage. It's actually a very, very stable molecule. It's evolved to be very unreactive. But nonetheless, there's so much DNA that damage to it is actually very common. And while you're listening to this lecture, you are suffering from thousands of DNA damage events in your genomes and many cells in your body, as am I. And you probably know that damage to DNA can cause a mutation, and that can lead to cancer. Luckily, we're really, really good at repairing our DNA. So although damage to DNA is very frequent, it is very efficiently repaired. And that means that almost all the damage that ever happens is reversed or fixed and patched up. So that's really essential for life to exist, and that's true for bacteria. It's also true for all living things, including humans. So that's a little bit about DNA, and I want to now look at the other main threat to living things, and that's viruses. So I don't think viruses need any introduction anymore. We're all sick of hearing about them, I think, after the last 4 years. But just to make it clear that it's not just humans that suffer from viruses. All living things are attacked by viruses, and that includes bacteria. Bacteria have got really cool viruses. So, I mean, this is an artistic image, but it's a fairly accurate one of viruses attacking a bacterial cell. And you can see the virus looks like a little moonlander in green there. It's got a head, and you can see the DNA in the head. And then it's got a sort of a tail or a spike and some legs. It uses the legs to grab hold of its host, and then it injects the DNA through a sort of needle that it pushes through the bacterial cell. And once that DNA gets into the cell, it can infect the host, and it can take it over, often kill it, and replicate more copies of the virus. So viruses are a sort of parasite. They're a genetic parasite. They can't live on their own. They need to infect a host to replicate themselves, and they need to use the machinery from the host to do that. And so there's a sort of battle going on between bacteria and viruses that is never ending, and it's probably been going on for as long as life has existed. As you might expect, then cells have evolved very good ways to defend themselves, and they're defending their DNA really against these viruses. And so if we think about the ways cells defend against viruses, we'll take the example of bacteria. So you may have heard of restriction enzymes. These were the first type of defense against viruses discovered in bacteria around about the 1970s. These are particular protein machines, enzymes that detect invading DNA and then just cut it up. So they detect a foreign piece of DNA, they chop it up to stop the virus replicating and taking over the cell. The restriction enzymes, when they were discovered, began to be used as a tool in molecular biology to allow scientists to cut and paste DNA and to make new constructs of DNA. And that's really the birth of molecular biology as a discipline. That has revolutionized the whole of this area of science because it allows us to create DNA and modify it in a very efficient way, in ways that are very useful. Now, in the last 10, 15 years, we've discovered there are more and more types of defense system in bacteria. One that I'll spend a little bit of time talking about in more detail is the CRISPR system, which was discovered in E. coli in 2008 and is an interesting form of adaptive immunity. And then in the last 5 years or so, our knowledge of bacterial genomes has allowed us to find many, many more different defense pathways. There's been a flowering of this area of research where we've realized that there are hundreds of different ways to defend against viruses. And the average bacterium probably has maybe 6 to 10 different types of defense pathway against viral infection. And that's roughly the same as us. We have probably about 6 to 10 different ways to defend against viruses as well. And what we're finding as we study these systems is that many of them are related to eukaryotic or human defense against viruses. And actually, the bacteria evolved these systems, and we inherited them from bacteria. So, for example, the simple molecule here, which is made by combining ATP and GTP, which are two of the common chemicals in cells. It's called the molecule is called CGAMP. That's a signal. It's a sort of distress signal when cells get infected by a virus, and it's used to activate defense pathways. And that's been made. It's actually been evolved in bacteria, and we have inherited that and still use that as a defense against viruses. Okay, so I want to focus in on the CRISPR system, and this is one of the areas that we actively study in our laboratory. So you may have heard of CRISPR. If this was a live lecture, I'd ask you to put your hand up if you'd heard of CRISPR before, and I'm imagining maybe a third to half of you have put your hand up. So briefly, I want to explain what CRISPR is. So as I said, it's an adaptive immune system. It's a really unusual system because it's a system that rewrites the genome of the bacterium, and basically CRISPR can capture a small piece of the invading viral DNA. It will incorporate that into its genome and form a sort of library. So these different colored bars are different pieces of viral DNA in the genome. That then is used to provide defense. So if a virus that's already been encountered infects the cell can defend itself because it reads this CRISPR sequence. It makes some RNA copies. That's called transcription. And it uses those to load an effector protein, and that effector protein then provides defense against the virus when it infects again. And so CRISPR is used by bacteria and archaea to defend themselves against viruses by degrading their genetic information. And it can constantly evolve and capture new sequences. So St Andrews actually has a special place in the study of CRISPR biology, because in 2013 we had the very first worldwide conference on genome editing with CRISPR, and this was a bit of lucky timing actually. This was just the year when the discovery of Cas9 as a molecular scissors had been achieved by two people who are highlighted here, Emmanuel Charpentier and Jennifer Doudna. And the first examples of genome editing had just been described. And so that summer, many very famous scientists from around the world came to St Andrews to talk about this. And this is just a group picture. The work that Emmanuel Charpentier and Jennifer Doudna did won the Nobel Prize in 2020. But it arose from this paper in 2012, where they showed that they could program a CRISPR enzyme Cas9 to cleave any piece of DNA anywhere in a cell. This led to a flowering of research in genome editing at breakneck speed, and in very few short years, by 2020, in fact, patients were being cured of genetic diseases. This is Victoria Gray, who was born with sickle cell anemia. That is due to a mutation in her DNA. And using CRISPR, using Cas9, scientists were able to reverse the mutation and give her a complete cure from her disease. And this technology is now being rolled out more broadly against very many different genetic diseases. I'll be happy to talk more about that if people have questions. But if a disease is caused by a mutation in DNA, it can potentially be cured using CRISPR to edit the DNA and to fix it. I just reiterate here before I move on that this very, very fantastic, powerful technology arose from very basic research, where the scientists were not really setting out to discover a technology. They were just trying to understand how a bacterium defended itself against viruses. Okay, so Now, I want to spend the rest of this talk talking a little bit about the research that we've been doing recently. And I want to introduce one more thing to you, which is the microbiome. And now this is something else you've probably heard of. If you've taken probiotics, you're feeding your microbiome. And your microbiome is the bacteria that constitute an important part of your body. We all have them. We probably have about 1 kilogram of bacteria in our guts mostly. They're also on our skin, of course, but in our guts, about 1 kilogram. It's about the same weight as your brain. And it's a really important part of our bodies for health. Roughly 100 trillion bacterial and archaeal cells in the gut, and probably also 100 trillion viruses. And these are different bacteria. The constitution of your microbiome changes with your age, with your diet, with your lifestyle. And we now understand that it's so crucial for human health. So we were interested in some of the bacteria that are found in the human gut, in particular an organism called Bacteroides fragilis. Since I can't pronounce that, I'm going to call it BFRAG for short. So BFRAG is very commonly present in the gut of all humans and many animals. It's normally perfectly a good neighbor to have, but actually sometimes it can cause disease. So it can cause pathology if there's a disease state. And we were interested in BFRAG because it had a very interesting CRISPR system that we wanted to study. So I'm going to briefly describe that. So I need to introduce a slightly different type of CRISPR defense. So Cas9 is a very simple form of defense that degrades viral DNA, cuts it like scissors. But BFRAG has got a different type. It's called type 3 CRISPR, and it works by making a molecule as a second messenger. This is a sort of alarm or SOS message in cells. So when the type 3 CRISPR system detects an invading viral RNA, it makes these molecules. It takes ATP and it makes them into rings. These rings can be 3 ATPs or 4 or 6 in size, and those molecules act like an alarm signal. Really, they activate the defense in the cell. The cell has a range of different defense enzymes, which when they bind these molecules, they then start degrading important constituents of the cell. For example, RNA or DNA. Now that provides immunity either by killing the virus, by degrading its DNA or RNA, but also by degrading the cell's own DNA or RNA, which sounds like a bad idea, but actually that slows down infection. It slows down the ability of the virus to replicate. So BFRAG has a really interesting type 3 CRISPR system, and we'd studied a lot of these enzymes in the past and published on them. And we turned our attention to the BFRAG system because it has a different type of effector. This is the enzyme, the protein that we think is activated by the signal molecule. And in this case, in BFRAG, it's a membrane protein. This is a predicted structure. It sits in the membrane and provides defense. We wanted to understand how that worked because it hadn't been studied. So this was work undertaken by a PhD student, Hao Tian, shown here in the lab. Hao Tian was a very good molecular biologist, and she quickly assembled the CRISPR system. So over on the left here, I'm just showing the proteins that make up this system. It's quite a complicated protein machine, but she could make this in E. coli and build it, and she showed that it was active in cells and it provided immunity. But she was stuck for quite a few months here because this enzyme, this CRISPR enzyme, just didn't function in a test tube. So we looked for the molecules, the signal molecules that were normally made by CRISPR, and they're just not present. So the BFRAG system just didn't seem to make any of these molecules. So it was a bit of a stumbling block, a real puzzle. So to solve this, Hao Tian took a step back and activated the whole system in E. coli, so in vivo in the bacterium. She activated the CRISPR defense. She then looked for new molecules that were made in the cell in response to the activation of the system, and she found a very nice peak. This is a peak that we identify by a technique called HPLC and mass spectrometry, where we can look at single compounds that we can isolate. And she saw this molecule is present when the system's activated, when it's absent in the control samples. And this molecule has a very specific mass. We can determine it very accurately by mass spectrometry, but it didn't match anything known in any catalogue of molecules that have ever been made or identified. So it was a mystery. So we're calling it here molecule X. So Hao-Tian now had to identify this molecule. And she did that by using mass spectrometry to break the molecule down into fragments. And this is a sort of jigsaw puzzle. So you break the molecule down, you measure all the fragment sizes, these are shown on the top here, and then you try to work out if any of them match things you know. And with a lot of hard work, it took about 4 or 5 weeks. Hao-Tian was able to piece together what the molecule was, and she finally cracked this sort of puzzle at 1 o'clock in the morning one night, and sent a very excited email that she'd found the molecule. And as it happens, I was awake thinking about it as well. So that's not a normal situation, but sometimes you get these very exciting situations in science. So this is the molecule. It's actually a new combination of 2 very common molecules, ATP, which I've introduced already, which is the energy molecule in cells, and another molecule called SAM. SAM is a way to pass small carbon units between different chemicals. So they've never been seen joined together, but this CRISPR system joins them together and makes a molecule that we call SAM-AMP. Okay, so it's a combination of SAM at the top here and AMP on the bottom. So once we'd found that, Hao-Tian was able to reconstitute this system in a test tube and show that when you provided the CRISPR system with ATP and SAM, now you could see the molecule SAM-AMP being made. So that confirmed that this really was the real signaling molecule of the cell. How we think this now works is that in these befrag organisms, they detect viral RNA when they get infected. They combine ATP and SAM, and they make this molecule SAM-AMP. And what we think SAM-AMP does is it binds to the membrane channel protein. That protein then opens a channel in the membrane and allows the cell's contents to leak through. So it makes cells leaky, and they lose important molecules. That's obviously kind of bad for the cell, and it might lead to the cell dying. But it's also bad for the virus. And if a cell is infected, it's actually better that it dies than allows a virus to complete replication. And this work is not yet published, but we're just waiting for that to come out. It's in press in Nature at the moment, which is it's going to be nice to see that come out. So just to finish up then, what are our next steps for study of this system? So I've shown you that this human microbiome bacterium makes this particular molecule. We understand that now, but we don't understand how it then activates the membrane channel protein to provide defense. And we're beginning to study that using structural biology and biochemistry, both in the test tube and also in cells, to understand what happens to a cell when this system is activated. We're really interested in whether this molecule is the first of its kind. It's actually a very difficult molecule to make by synthetic chemistry, but we can make it with the enzyme, and we can modify it by using different building blocks. So this is the first of a family of molecules that we think will be bioactive and might be useful as drugs. And of course, we can't forget about the viruses. So the viruses, of course, are very good at overcoming the defense systems that bacteria have, as well as human defense systems. And so what we expect is that the viruses have got ways to break this molecule down. We've discovered other viral enzymes that degrade the other signaling molecules that we know about. And so we think the odds are that there will be viral enzymes that break this molecule up before it can be used to activate defenses. And so that's another area where we're starting to look for some interesting new enzymes. So I'll finish there. Just thank to all the people, past and present, from the lab. and to our collaborators. We have many collaborators in St Andrews and further afield, without whom this work wouldn't be possible. And finally to the funders. So my lab has been funded ever since I've come to St Andrews by the BBSRC Research Council, and in the last few years by a grant from the European Research Council. So thanks to all of them, and thanks to University of St Andrews for having me here for the last 23 years. And I'd be very happy to try to address any questions you might have. Thank you.
Well, thanks to you, Malcolm, for what's a fantastic talk. It's been really genuinely enlightening and interesting, and that's been evident in some of the questions that are coming through as well. If you're watching at home, do please continue to ask your questions. I know there's a lot that you'll be wanting to ask, because there's a lot in that presentation, but please do just keep them coming in, and we'll get through as many of them as we can. I was just going to kick off with one of my own first. Something that I picked out in your talk was you showed that great slide of the viruses attacking, and it's easy to kind of imagine that as a kind of a battle between two kind of sentient beings. But I just wondered, do you consider viruses to be alive? Are they conscious or sentient? Or...
It's a good question. It's a bit of philosophy, and it does divide opinion. Some people are very strongly feel that viruses are not alive. Personally, I think it's not very helpful to consider whether are they alive or not. To my mind, they're a biological entity that can reproduce, but they need help. So they're a parasite, so they can't reproduce on their own. But that is true for many other forms of life, which need to parasitize another host in order to replicate. So to my mind, they are basically, I would say, alive, in the way I think about it, and which is probably in sort of a reductionist, mechanistic terms.
They don't have hopes and dreams.
I don't know. Do they have less than bacteria? I'm not sure.
And I also picked up on, you said there's something like 6 to 10 ways that we as humans have of protecting ourselves or protecting our DNA from damage. Could you talk a little bit more about that, and what are the specific ways that we do that?
Yeah, so lots of them are, as I said, are inherited from bacteria. So we now, funnily enough, we found them first in humans. I guess we're quite, motivated to look for that. But we then find that actually they've come from bacteria. So there are similar ways to the ways I've described. So usually there's an important way to detect the infection has happened. That's the crucial first step. Often that's by detecting the virus's DNA or RNA. This is true for SARS-CoV-2, for COVID-19 infections, for example. Once you detect the virus, you can activate a defense system, and very often these result in cells dying. So it makes sense for a cell to die if it's infected. That kind of makes sense if you're a lung cell, for example, because there are billions of lung cells. But even it makes sense for a bacterium to die, and that's a little bit more counterintuitive, because once you're dead if you're a single cell. But your neighbors are related to you, so at the level of evolution, that tends to make sense. So in humans, we will degrade the viral DNA or RNA by activating a nuclease, a bit like the CRISPR nucleases, or we might puncture a hole in the cell membrane in the same way that I described for the BFRAG system. These are different ways to kill cells. We also have systems that signal infection to other cells. So interferons being the most obvious example. Interferons send a message, a warning to other cells. There's a virus around, get ready to defend yourself. And interferons are something that have been evolved or in animals rather than bacteria. So bacteria don't get all of the plaudits for immunity. The other big system that's important in animals is, of course, the antibody system, and that's specific to more complex eukaryotic cells. So bacteria don't have an antibody system, but it's an adaptive system, and analogous to CRISPR, which is also an adaptive system, although they're not related at all.
Yeah. Is there potential for using viruses to kill harmful bacteria? Is that, can we harness the power of a virus to do something that we want it to do?
Yeah, I mean, that's actually an area where there's a lot of resurgent interest. Viruses have been used to kill harmful bacteria for over 100 years. Before antibiotics, people were using viruses, and it can be very effective. So it has actually some advantages over antibiotics, because a virus that infects a bacterial cell is very specific. So you can have a virus that only kills the bad bacteria that you want to kill. But you don't want to kill your own microbiome, right? So all those well-behaved bacteria in your gut, I mean, you know what happens when you take antibiotics. Your health does suffer because you kill too many bacteria. So viruses can be very specific, and they are now being used to cure diseases. And this is particularly now important because antibiotics are starting to run out. We don't have a lot of new antibiotics on the shelf, and bacteria have evolved resistance. And so there's now actually a lot of interest and a lot of money going into the development of viruses, or phage, or bacterial viruses are called phage. Phage therapy is the use of bacterial viruses to cure infection. And it's definitely it's a big area where there's a lot of excitement.
Great, thank you. And final one for me, and then this kind of goes into a couple of questions that people have already asked online. I was really interested when you spoke about the use of CRISPR to tackle sickle cell, and how that's actually can cure people completely of the condition. Are there other what's the future of that? And someone's asked specifically about cystic fibrosis and whether CRISPR can be used to correct the mutations in CFTR. But could you talk a little bit about the kind of future of that technology for specific conditions? And as a kind of an addendum to that, are there any particular dangers concerned with doing that in live humans?
Yeah, absolutely. So CRISPR has really changed our ability to do genome editing, and that allows us to correct lots of, in theory, any mutation that might cause a disease. And many diseases are caused by mutations in your DNA. Often they're inherited. So cystic fibrosis is one of the more common ones. And absolutely, CRISPR is being used and developed to treat and hopefully cure cystic fibrosis. And there are clinical trials underway. It turns out that the actual genome editing is actually the easier part of this technology. But the difficult part is sometimes just getting the CRISPR system into all the cells, into enough cells, particularly in an adult human. How many lung cells do you have to edit to relieve symptoms of cystic fibrosis? That's a very large number of cells. So there are different delivery mechanisms, and actually the most common is to use a virus. So a virus that infects human cells, you can program it with a payload of a CRISPR system that's programmed to attack the mutation and correct it, and then you try to maximize the number of edits of the defective cells to make a cure. So this is being rolled out to tackle lots of diseases. They're mostly in phase 1 and 2 clinical trials, although there are several examples where patients have been cured of a variety of diseases. The one of the limiting factors is the expense. So it's still expensive. It's not a treatment that can be rolled out, for in a cheap way, let's say. There are also, as you hinted, potential risks. You don't edit a genome, casually. You have to be very careful. You have to be very sure that it's safe, and there are risks. There's a potential to cause an unexpected change, which might lead, for example, to a mutation in cancer. And so it has to move slowly. It has to be checked every step of the way to understand the safety. Of course, you might be trading off a non-zero risk against a very serious disease, and so the balance there is very variable, but an important one to think about.
Thank you. Got a question from Dan, who's asking about in the analysis of the structure of these kind of complex molecules, how important or not is the power of modern computers? And I guess as a bit further to that, I know AI is obviously expected to be very important in drug discovery. How much is that going to impact on the kind of work you're doing as well?
Yeah, I mean, that's a good question. Yeah, the quick answer is absolutely vital. The slightly longer answer is that, I mean, several of the structures I showed you are generated by AI. So there's a new development in structural biology, which is called AlphaFold. It's developed as an AI method to predict the structures of these proteins and protein complexes. It is revolutionizing structural biology in several ways. It allows us to build a model that is often very useful, although it's a model, but often relatively accurate and useful to allow us to progress science more quickly. We have to confirm, these models and predictions. When we do confirm them, often they're basically right, at least in my experience. For drug discovery, it's a bit more complicated. You really still need to solve the actual structures of targets, and then hopefully solve them in complex with the drug bound inside. And that's not something that AI has really revolutionized yet, although progress is now very, very fast. So I expect this to continue to accelerate the progress we can make.
Thanks. We've got an anonymous question that's asking about, you said earlier that it's better for a cell to die if it's infected so as not to continue to replicate viruses. And someone's asked if you anticipate a similar mechanism for cancer cells whereby a transformed cell undergoes apoptosis so not to become a tumor.
Well, that's right. That's exactly what's happening there. The logic is the same, that if a cell has lost control of itself, some of the checkpoints that keep it replicating in a normal programmed way, then there are safety mechanisms built in which have evolved to sort of force that cell to commit suicide in order to avoid becoming a tumor. So the parallels are very strong there, and the evolutionary theory in terms of cancer cell, both cancer cell growth and how cancer cells develop immunity to the drugs we use to treat them. That's very similar to the way that bacteria develop immunity to the antibiotics, for example, driven by evolution. And these safeguards that cells have where they may self-destruct to protect the organism are essentially very similar. There are a lot of parallels there.
Just we're running out of time, but I was just going to ask as a final question to you. Just what's the future of your? I mean, it's obviously very exciting, the stuff that you presented today. But what's the future of your research and your kind of collaborators at St Andrews and beyond? What's the next big horizon that you guys are looking at?
Well, I mean, I think this whole field has exploded, and there are now. It's a bit like a playground. There are hundreds of systems. We need to understand how they function, and as a biochemist, I like to take them apart and put them back together and understand how their mechanisms. And I guess the next step is to understand really how these different systems interact with one another. So you need, let's say, an average of 6 different systems, but how do they communicate? Is there communication? Are there ways in which multiple pathways provide us a multi-layered defense that is better than the sum of their parts? And so those are the areas we're starting to investigate, and that brings its own challenges as it's fundamentally more complex. But it's, I think, also relevant for the human immune system, where you have several different levels of different defense system which interact to different extents.
I'm going to ask you one last question that an anonymous person has put in, and they've obviously paid a lot more attention than I have because they've grasped the SAM GMP bit. But they're saying, have you seen evidence for SAM GMP as well as SAM AMP? If that makes sense.
That's a good question. We can't make it in the test tube, so we can try to do that, and we can't make that, but that doesn't mean it doesn't exist. these different signal molecules, we now know of dozens of them. And the reason there are so many is probably because the viruses keep attacking them, right? So, so if a virus finds a way to degrade SAM-AMP, it's quite possible evolution will develop a different molecule, and that could be SAM-GMP, right? If that stopped the virus breaking the molecule. So I wouldn't rule out anything at the moment, but yeah, right now we haven't seen it yet. But it's a good question.
Great, thank you very much. That was really genuinely enlightening. So thanks again for a really thoroughly interesting talk. It's great to get these insights into the research and the passions of our academics. So it just falls to me to thank Malcolm once again profusely for taking the time to talk to us on a subject about which he's clearly passionate and world leading. I can tell from the volume of questions that have come in that it's really struck a chord with lots of people. So thanks very much for taking the time to speak to us. Thanks also to all the staff who've given their time to make the arrangements for this event. There's loads of work that goes into these, and it's very much appreciated by all of us. And finally, a big thank you to all of you, our esteemed alumni and students and staff and friends for attending this talk. We're delighted to be able to welcome you virtually back to St Andrews every few weeks, so please do continue to attend these events. As I said, our next talk is on the 15th of November, and that will be Professor Julia Priest from the School of English. But aside from that, we look forward to seeing you again then, but thanks very much for joining us and enjoy the rest of your evening.
Thank you. Bye-bye.